Hi everyone, thank you for tuning into my talk, Hello Darkness, Red Algal Patch Dynamics on Temperate Rocky Reefs, which I am presenting on behalf of my co-authors, Tristin McHugh and Matt Edwards. Kelp forests are ostensibly some of the most diverse and ecologically important marine habitats worldwide. This is PSA after all. I don't think I need to convince you about the wonders of kelp forests. But for this talk, I'd like you to think about that three-dimensional structure made by canopy-forming species, a view from the benthos where red algae are found, and the kind of environments found under all of this structural complexity. From the literature and our experiences in the water, we can see that kelp forests are organized by distinct vegetative layers: canopy, subcanopy, understory, red algae, and encrusting. Here, we're focused on this red algal layer, which we generally think of being shaded by canopy-forming species. Each layer has the potential to shade the layer below it and can exert a direct negative effect. Disturbances can remove some of these canopy-forming layers, which can alter light levels and alter community composition and abundance within kelp forests. So, how do disturbances alter kelp forest ecosystems? Well, we can have a weak disturbance event roll through the system and remove wave-susceptible species such as the canopy-forming layer, but leave most other species intact. Or we can have a stronger event roll through the kelp forest and remove many individuals throughout these layers. However, red algae have been observed to be quite resilient to storm events. So, does making the distinction between the frequency and magnitude of storms matter? Starting around the 1980s, researchers in California conducted experimental manipulations of kelp canopies to investigate how understory species, and not really red algae, respond to disturbances. They typically did consistent removal of kelp canopies and found an increase in understory species, formerly suppressed by shading layers, opportunistic species recruited to these new highlight environments, and that algae may have fixed highlight and low light adaptations. And so all of these findings led us to the idea that light may be a major driver of understory algal community composition, which is no brainer. However, not all of these studies actually tested light adaptations in the algae they were studying. However, Clark and others did classify red algae in experimental plots in Carmel Bay through various combinations of canopy removals. They then organized red algae into light requirement categories based on their relative percent cover within their canopy removal treatments. Although they grouped red algae into these light requirement categories, it is important to note that most species abundance across treatment varied only by a few percent, and that red algae were grouped into these light requirement categories based on community assemblages and not light adaptations or their physiology. Therefore, a big question that came out of this study is, do these differences in photophysiology drive community composition? Well, to answer that question, we conducted experimental removals in two disparate regions along the California coast, north and south of a major biogeographic break, shown here, the blue line. Within each region, we have 3 sites or blocks, and within each of these 3 sites, we had 4 treatments varying in their level of disturbance. Treatments were designed to resemble storm disturbance events of varying frequency and magnitude. Divers would get a weapon of choice, such as a kitchen knife, to simulate said disturbances. An infrequent strong plot simulated an annual disturbance, a frequent strong plot simulated seasonal disturbances, and a frequent weak plot simulated seasonal disturbances, but of lesser magnitude, and of course, a control plot to compare them against. This allowed for a nice comparison of both frequency and magnitude. All treatment plots were seasonally sampled using random point contacts to capture the percent cover or abundance of benthic placeholders. And from these data, we figured out who the most abundant red algae were. These algae were then used to understand algal light adaptations via photosynthesis versus irradiance curves, of which many of you are likely familiar with. We can increase irradiance on the x-axis to see a response in carbon uptake seen on the y-axis. Our photosynthetic parameters of interest are Pmax, the maximum rate of photosynthesis, and alpha, the photosynthetic efficiency. To capture individual species light adaptations, we ran between 7 and 10 species within each region seasonally. And for each species, we ran 3 replicate curves. A replicate was gradually exposed to 8 different light levels over an 80-minute period, and we recorded the amount of oxygen produced or consumed. Note that we did not run curves to saturating irradiances, and instead, using methods from another study, used light levels derived in situ. Now onto our results. I'm going to report our results based on region separately, starting with Point Loma in the south. Here we see our communities oriented in multi-dimensional space, separated by sampling period. These NMDS plots show our assemblages across all treatments, and clearly there's no real pattern. Our plots group out by time, but treatments all look similar to one another. And we can see this for each of our individual species plotted as bar graphs. The x-axis shows our different treatments grouped by sampling period with abundances on the y. We see that many species are rare, showing variation but no consistent pattern through time. We used a permanova to test for the effects of selected variables on red algal communities to look at differences in community composition, while treatment, sampling period, site, and their interactions were significantly different from one another. Take a look at these residuals. They explain nearly 72% of the variations seen in Point Loma. Wow. Still, we are interested in algal photosynthetic parameters after all. After running our P versus I curves, we saw no difference in overall oxygen production, seen here on the y-axis, across our light levels, seen here on the x. However, we do see big differences in their photosynthetic parameters, which is definitely to be expected, but it does not appear to be a driver of their assemblages. Red algae are different from one another after all. Regardless of the differences in light adaptations observed, our results signify that red algae do not exhibit differences in light adaptations that can be used to classify their benthic community assemblages. And light adaptations do not appear to be strong enough to characterize red algae as having high light or low light adaptations within Point Loma kelp forests. Moving on to Carmel Bay in the north. And spoiler alert, we observed similar trends as we saw in Point Loma. So I'm going to move through this section a little bit faster. Again, we see no clear grouping between treatments in our algal communities through time, though there are differences between our sampling periods. A plot of abundance versus treatment shows that, just like in Point Loma, many species abundances change through time with no clear pattern across treatments. A permanova for Carmel Bay showed similar results to Point Loma, except that the residuals are even higher, explaining 81% of the variation we saw in this study. Oxygen produced by our reds looks similar, except that some species may be more productive in summer months, but that wasn't addressed in this study. And just like Point Loma, we see big differences in photosynthetic parameters for red algae in Carmel Bay. But again, these differences cannot explain patterns of abundance in our plots. Red algae are different from one another. And so to summarize, neither the frequency nor magnitude of disturbance appear to significantly alter benthic red algal assemblages. And these responses were consistent in both Point Loma and Carmel Bay. We did see differences in photosynthetic parameters, but there's no evidence to suggest that red algae can be classified as high light or low light adapted, and that these light adaptations were not enough to influence community assemblages. So what does drive community assemblages on temperate rocky reefs? Well, variation in abiotic factors such as water temperature and nutrient availability, grazing pressure from encroaching urchins, anthropogenic disturbances such as ocean acidification, and a whole host of other factors. Basically, it depends. There are a lot of folks that helped make this study happen. We'd like to thank everyone involved for their immense help in the field and in the lab. And of course, funding was graciously provided to facilitate this study. And thanks for tuning in. Please feel free to reach out. I love talking about all things phycology. How do I stop it recording? Help, help.